So in this unit, we're going to start talking about cells and the cycle that cells go through throughout their lifetime. We're also going to tie this into cancer and how cancer is caused by some mishap within the cell cycle. So the cell cycle is broken down into three main steps. The first part is interphase, and this is where the cell spends about 90% of its time. And a way to think about interphase is the part of the cell cycle where the cell is doing its everyday function. Interphase is broken down into three smaller subphases. The first phase is the G1 phase, and that G stands for growth. And during the G1 phase of interphase, the cell is obviously growing. It's preparing for DNA replication, which is making a copy of DNA, and it's performing its everyday function, so whatever the job of that cell is. The second part of interphase is the S phase, which stands for synthesis. This is the part where DNA replication occurs. And remember that DNA replication is just when DNA makes a copy of itself as it prepares to divide into two new cells. And following the S phase, there's going to be a G2 phase, and this stands for growth 2. So the cell is going to continue to grow, and it's going to start making its final preparations before division or mitosis, which is actually the second part of the cell cycle. This is one of the shortest parts of the cell cycle, and this is when the nucleus divides. So the DNA forms into chromosomes, and those chromosomes are split. And then following mitosis is cytokinesis, which is when the actual cytoplasm of the cell divides. In the next video, we'll talk a little bit more about what mitosis actually is. One thing that's really important to understand about the cell cycle is that there are built-in checkpoints. And these checkpoints are composed of proteins or enzymes that ensure that everything is happening properly as the cell divides. So there are three main checkpoints in the cell cycle that we need to be familiar with. The first is known as a G1 checkpoint. This is the checkpoint that occurs right after G1 before the cell enters the S phase. And this is just describing what happens there. So it's checking whether or not the cell is big enough for the synthesis phase. It is also checking to make sure if there's any damage in the DNA. If there is damage in the DNA, in the G1 checkpoint, there's a really important protein called P53. This is a protein that's responsible for checking the DNA for damages and then trying to repair it. If the DNA is not ready to continue on to the synthesis phase, either something has gone wrong or the DNA is damaged, it's going to go into what's known as a G0 phase. And that's a resting period so that the cell can catch up or be fixed or do whatever it needs to do, and then it can come right back into the cell cycle. The second checkpoint is what's known as a G2 checkpoint, which occurs right after the synthesis phase. And so this is going to make sure that the DNA was replicated properly during DNA replication. And if it was, it'll continue on into mitosis. And then we have another checkpoint in the cell cycle, which is known as an M checkpoint. And this is going to occur right in between metaphase and anaphase of mitosis, where the chromosomes align in the middle of the cell. The M checkpoint is responsible for making sure that the centrioles, which are those little strings, or the spindle fibers, excuse me, which are those little strings that come out of the centrioles, attach to the chromosomes properly. We'll talk about that a little bit more in detail in the mitosis video. Here are just a couple of diagrams showing some important cell processes. This first one here is called apoptosis, which you may recall is programmed cell destruction. So throughout those checkpoints, sometimes a cell is beyond repair. And so what the cell cycle and those proteins and those checkpoints can do is send a cell into apoptosis, or tell the cell to essentially explode or self-destruct. And so this is a general diagram of what it looks like. You start with a normal cell and you end with what's known as apoptic bodies. Here's an example, an SEM of what a cell undergoing apoptosis looks like. It just starts forming a lot of blobs until it essentially disintegrates. And this right here is showing you what the P53 protein looks like. It's a couple of polypeptide chains, which means it has a quaternary structure, and this is the DNA molecule. So if there was damage here, the P53 protein attaches to it and then makes any repairs. Real quickly, I just want to talk about what are some of the major differences between cancer cells and normal cells and how this ties into the cell cycle. So cancer cells are non-differentiated cells, which means they don't have a specific job. They just continue to grow uncontrollably without any checkpoints. They normally have abnormal nuclei, and within those nuclei, they contain an abnormal number of chromosomes. Because cancer is uncontrolled cell division, most cancer cells have way too many copies of chromosomes because DNA replication happened much quicker than the cell could actually divide. They do not undergo apoptosis, which means that p53 protein, which is responsible for either fixing the DNA or telling the cell to go, self-destruct, cancer cells do not respond to that. Either the p53 gene is missing in those cancer cells, or those cells do not respond appropriately to the p53 protein because the gene is turned off. There's no contact inhibition, which means as cells normally clump together, they most of the time shut off. Cancer cells don't do that. The more they clump together, they'll still continue to grow and grow and grow. They're obviously very unorganized and multi-layered, which can lead to a tumor, and they can undergo metastasis and angiogenesis. Metastasis means spreading, so tumors can spread throughout your body, and angiogenesis means when a tumor literally takes a blood supply and hooks it up to itself. Cells need nutrients to divide, and so angiogenesis is a way to ensure that those tumors get all the nutrients they need to continue to grow. Normal cells are practically the complete opposite of that, and so you can see here some of those differences. Lastly, I just want to talk about some big terms when we talk about the origin of cancer. These terms may sound familiar to you, but the three I want you to be most familiar with is right here. The first is a proto-oncogene. These are genes that are going to promote the cell cycle, and typically these are your normal genes. So all of our chromosomes are going to have these proto-oncogenes, or those normal genes that just tell a cell to continue going through the cell cycle. When a mutation occurs in those proto-oncogenes, you can actually have what's called an oncogene, and these are those cancer-causing genes. And a way to think about these oncogenes is like a gas pedal in the car with no brake system. And so these genes tell the cell to keep going through the cell cycle, but they never stop. And so that's where you get the uncontrolled or the rapid division of cancer cells. You also have these tumor suppressor genes, which are genes that are responsible for suppressing tumors from occurring. And the most common ones are p53 gene and the RB gene. And these two proteins, p53 protein and RB protein, are responsible for checking those cells and making any repairs before it becomes cancer. And one thing that's really interesting and kind of new with cancer research is this idea of telomeres. So telomeres are the very ends of your chromosomes or your DNA that don't code for anything. They're just there. They're a bunch of random letters. And so what happens that every time your DNA replicates, those telomeres shorten. And over time, once your chromosomes have used up all of their telomeres, those cells stop dividing. Well, there have been recent studies to show that the enzyme responsible for shortening those telomeres, known as telomerase, isn't working or it's mutated. So the telomeres are actually not shortening, which means those cancer cells keep dividing and they never stop. And so a lot of current cancer treatments are targeting telomerase and those mutated genes that cause it.